Okay, I'd like to start by thanking the organisers for giving me an opportunity to come here and talk to you today about Neuro AI. Now, Neuro AI is something I and many others believe is going to be a catalyst for a whole new generation of AI systems. So I want to put forward initially the case for Neuro AI, why we think that it's going to replace, or not replace, but it's going to catalyst a whole new generation of AI that's more brain-inspired. No, can everybody hear me? Okay. I'll stay back here. So, case for Neuro AI. So, have you listened to AI people, or you listen to podcasts or YouTube clips of talks about AI? They always talk about, the aspiration to get towards human-level intelligence and to get towards human-level intelligence with acceptable power requirements. But is that really going to be the case, given the direction of travel of AI research? AI research is not and is unlikely to be conscious of the world around it, to sense the world around the way we do, and to take action. And it cannot work with small data sets. It works with huge data sets, thousands and thousands of data sets, even just to distinguish between an apple and an orange. So, I don't know why that's flicked on. Anyway, so even with scaling, AI is unlikely to even reach the intelligence of even a small animal. And the power requirements will be unacceptable. It's forecast for generative AI that it will use 1.5% of the global electricity demand over the next 5 years. That's huge. That's absolutely huge. So what's the solution to this? I'm not saying to replace the direction of travel of AI, but there's something else we can add in there, another ingredient. And if we look at what we're doing in the minute, the networks that we're using, whether it be generative AI, whether it be networks we use in the lab, they all use a single type of neuron, an oversimplified wiring. So they're all wired together in a very, very oversimplified way. If we look there at the graphic on the far side, apart from inhibitory and excitatory cells, there's lots of other cells in the brain. There's interneurons, there is motor neurons, there's sensory neurons, and even within those families, there are different kinds of neurons. So this idea of cell diversity we don't use in current AI, but it's there, and the neuroscientists know about it. Because cell diversity underpins intelligence in biological systems. I know about this because I work very closely with neuroscientists. So there's a need to understand cell diversity. There's a need to understand wiring diversity and embed these ingredients in AI. And that's where Neuro AI comes from. That's part of the driving force behind Neuro AI. So the problem with that and doing that sounds okay, and I do some of that because I would, if I had PhD students, I would bring in neuroscientists to help me supervise them. So they're learning from neuroscientists as well as learning from me. I'm a computational neuroscience. I don't work on the experimental end of it. But the AI community at the minute is made up of computer scientists, engineers, people like that. They lack the neurobiology training. So that limits their ability to draw from neuroscience. So the way to overcome this, this knowledge gap, is a whole new generation of researchers who are trained to be fluent in these combined areas. Just jumping on again. These combined areas of experimental neuroscience and computational neuroscience, or experimental neuroscience and neuromorphic computing. That is where we need to progress and focus our learning on. The graphic on the right shows why. At the left-hand side, you've got the experimental neuroscientists. On the right-hand side, you've got the AI people. But the AI people find it difficult to talk to the neuroscientists. I'm a computational neuroscience. I've worked closely for years and years. I'm only beginning to understand their papers. I'm only beginning to understand the way they present their data. They just speak a different language. So it's not surprising that AI has lost its way and hasn't drawn from neuroscience for a very, very long time. The AI algorithms that are currently used, whether it be generative AI, whether it be AI we use in the lab, like McDaid, Resume, all of them, they're all based on what we knew about the brain 20, 30, and 40 years ago. They bear little or no relationship to what's happening to the brain. So it's nearly impossible to aspire towards human-like intelligence. So Neuro AI is a research field situated right at the intersection of experimental neuroscience and AI. It provides the link. It'll tie neuroscience much closer to AI. So there's a pressing need to invest in fundamental neuroscience in AI. So the other part of the case, I suppose, is there's a vast amount of knowledge that already exists about the brain. I'm going to show you some of the things that's been done, that we've done ourselves with others, functions that we learned in the brain that are now available and could be easily transported and mapped into AI. So that amount of knowledge that already exists inspired the move to neuro AI research. It was part of the reasons why people started to go back and say, let's go back to basics and look at the brain again. So if we can do this, then neuro AI will become a critical driver and a source of inspiration for a whole new generation of AI, and it'll lead to energy efficient computing. We can do that. Even I, as a company, it's neuroscience, have got functions that are small networks that can do the same function as much large networks. I don't know why that keeps jumping on, but no, it's not me. So energy efficient computing, the brain consumes 2% of the body mass, but uses 20% of the body's energy. So that's equivalent to a 20-watt light bulb burning in your head 24 hours a day. It's not much when you think about what the brain can do in comparison with conventional AI, the energy it uses, and what it can do. So it can give us much more human sensory response capabilities. So neuro AI inspires new algorithms that match the perceptual and motor abilities of animals. And we think if we go back to neuro AI, we go back to basics, we can get there. And that then, there's a smaller step to human level intelligence. Okay. So I mentioned earlier about cell diversity. What I mean by that? Well, the different neurons wired together, we know will optimize computational function. For example, on the left-hand side, you've got a conventional neural network. It's got all the similar same types of neurons in there. It might have an inhibitory and an exhibitory, but it's similar to the neurons, the way they function. If you go back to the brain and look at the placement of different types of neurons, of your sensory neurons, of your interneurons, of your motor neurons, and other types and different types of them, there's families of them, then we can get a much more compact computational function that's much more energy efficient and takes up less room if you put it down on hardware. So that's cell diversity. And the abstraction level, where do we need to work? Well, to go back to basics, I don't mean to go back to the brain or go back to neurocortical columns. I mean to go back to single neurons and even ion channels. And this is where the neuroscientists want to work with us, down at the level of cells. They can probe channels and they can understand how channels transport ions in and out of cells or how they communicate with other cells. What they can't deal with is what happens if we change the expression level, the density of these transporters and channels. That changes the whole signaling pathways between different cells. That is what we want to get at and then move forward from there. And I can show you we've done that, and I can show you how that relates to AI. But that's the level of abstraction, single cells and channels that we need to be working at down there. So this is what we did know about synapses. You've got, I don't think there's a light in this. No, okay. So this is a presynaptic neuron, and we know that information travels down the axon, synapses onto the postsynaptic neuron, and there, that inset is a synapse. And what happens is the voltage changes in there, that's a spike. Calcium flows in from this cleft region, which is the region in between these two here. That then, those red ball things with stuff inside them, these are called vesicles. They're full of neurotransmitter. It moves, this calcium they move forward to the memory dock, and they spill out neurotransmitter into the cleft, and that moves across to the far side where there's receptors, and that binds receptors, and so information is gone from the presynaptic neuron to the postsynaptic neuron. That's our current thinking. A lot of our networks are built around that paradigm. That's not true, and particularly not true in areas like the hippocampus, because there's a new player in town now. It's called astrocytes, and one of the glia families. And these astrocytes are known, if you look over here, to reach out and then wrap the synapses. So there's a long thin process wrapping around that synapse. That happens an awful lot in the brain. Half the cells in the brain are glia cells, and half of those are astrocytes. So if you imagine a synapse like that, these processes come out, or they wrap themselves around it. And the reason that they do that is because they want to compartmentalize the synapse. They want to make it a compartment by itself. So it takes control then of species like calcium, sodium, potassium. And these processes are very thin. They're down to about between 150 nanometers. They're that thin that the neuroscientists can't probe them because they're just so small, and they push away. They're all very spongy-like. So they rely on us modelers to model them. And I'm working with somebody at the University of Liverpool trying to model at the minute. And there's some interesting features arise which will change the dynamics of how these cells interact with neurons, which will change the computational ability of that interaction. I'll come on to that later on. So these astrocytes enwrap about 10 to the 5 synapse, and they connect to multiple neurons, between 6 and 8 neurons. So they have got a finite domain of coverage, but there's lots of them, and they talk to each other. So there's a high level, you've got your low level neurons with synaptic plasticity, you've got high level of astrocytes and glial cells, which gives you network level plasticity, which we never consider when we talk about AI. The connection between astrocytes and neurons is named the tripartite synapse. That was coined about 15 years ago. And that's because there's another terminal, there's the astrocyte terminal now, so there's 3. And we've described the interaction between astrocyte and neurons. We've been building models since about 2010, and we call them the astrocyte neuron models. And I'm going to talk about them now, because the stuff that we've done in here, not us, but with others, could be immediately transported to better equip AI to deal with more complex functions. Just so you know that I'm being honest, this is an image of a synapse. The bouton is the end of the presynaptic terminal, the spine is the beginning of the postsynaptic terminal, at the dendrite, and the region between the dots is the cleft. And this yellow region is your astrocyte end foot, which comes out and it cradles around the synapse, wraps itself around, they call it cradling. And you can see there, it's not so easy that some of that yellow fade, that's going underneath, some of the yellow is going over the top, so it's wrapping itself around the synapse. When that synapse is active and continue active, they move closer and wrap themselves around it because they want to compartmentalize them and control them and use them. And I can show you some of the things we've done with that. So we tried to model this back years ago, and we worked with neuroscientists on this. It's not a very, I'm not going to talk technical here, but I just want to show you some things that arose out of simulations that we've done in conjunction with neuroscientists. So this is our synapse. This is not a very good diagram, but this is how we show this cradle or the end foot. And this is the wrapping round here. That's known as the synapse. We don't need to put a cleft up there. And on the far side, this is the thin process. That's the astrocyte body. But over here, you can see this region here, around there. It's this region in green. And on that membrane, we allow the expression of different types of transporters and pumps and channels for potassium, sodium, or calcium. This is the synapse. We allowed the different channels that appear there. But what we didn't allow is much of the flow of ions up and down the process away from the synapse or back to the synapse again. We know, because they work at Liverpool and others, that's semi-isolated from any other regions. So we only allowed ions to trickle down there. And what we got from that is this function here. I'm not going to explain any of this except to show you that whenever there's a spike, there's potassium released out of the synapse into this region. We must get it out of there right away. And the conventional wisdom was that that's taken up into here and then transported away. That's not right. We've shown that's not right. Neuroscientists didn't believe that was right, because that's not a very energy efficient way of doing things. So basically what happens is this potassium is stored in here, and then it's given back to this region and back to the synapse again for subsequent spikes. And this is the buildup of potassium piece here as you get different firing frequencies. The higher the firing frequency, the more spikes, the more potassium is released, the more potassium has to be stored. And so the height of this potassium concentration increases in that curved region. And that is really, and this graph here shows you what I really mean. This is the 0 level here. So going negative is potassium going into this region, and going positive is potassium leaving. And you can see it soaks it up, stores it, and then gives it back. That is applications for AI. Okay, so I'll come on to something else. This is the plot thickens here, because now we've got, I mentioned interneurons earlier on. These are GABA interneurons, and GABA is known to quiet everything down. If we GABA neuron, it'll quiet it down, it won't fire, it won't spike. But as you increase the frequency of the GABA neuron, or the presynaptic neuron, the astrocytes express receptors for GABA. They're called GABA-B. What happens there is they release a secondary messenger called IP3, and 5 minutes left. Okay. And that releases basically out of this calcium store a feedback signal, which changes a parameter called the probability of release, and that increases. So that means that the probability of release is low, even though there's lots of spikes, very little neurotransmitter will be released, so there'll be no learning. If the probability is increased, as it would be in this case, lots of neurotransmitters released, and you get learning because they bind to receptors, and more receptors are trafficked to that side, and so they can receive much more of the neurotransmitter, and the signal crossing is much stronger. What happens is, if you get to too high a frequency here, after a period of learning, there's two AG messenger releases called endocannabinoids. And they bind to receptors here, and they do the same thing, but as the level of IP3 increases, it stops. It shuts itself down. And that is a really useful function. And so, but basically at this side here, this is your STDP learn function. You take that to there as A naught. That function opens up to there, and it shuts down when the one stops learning. So here, as the probability goes above PR dash, it does it 2 or 3 times. You open the window, you learn. You close the window, you open the window, you learn. Close the window, open the window, you learn. So what does this give us? It's all very, it gives us this. AI at the minute, you have to tell it what to learn, you have to tell it how to learn, you have to tell it when to stop. It doesn't do it by itself, it should do it by itself, but this does it for you. Because every time the window opens, it learns a bit more by the map between the input and output, and it learns a bit more. When it's learned the map between input and output, it knows to stop. That's a huge advantage for AI. If we could build that into AI algorithms, and we can do this now. I'll show you a project we did with the University of York. We took these two conditions, and we modeled the probability release. Low frequency is low, as you get to higher frequency is higher, and then it goes back down low again. So it looks like a Gaussian function. So rather than building all them interactions, we're able to capture all that in a single Gaussian function. We built networks. We went to the University of York, and we got a proposal funded by EPSRC on robotics. And we built this network, we built it to control a simple robot. So these are all about the synapses. This is the move forward, turn right, turn left, and move backwards. So the robot had to basically do a simple task called obstacle avoidance. I'll show you what it did. This is a Pi robot at the University of York, their lab. So this is the learning phase. You can see here the firing rate increasing, you see the weights increasing. What you see is when it's learned to avoid all the obstacles in any type of obstacle avoidance system, it then everything stabilizes. It's learned it. What's the advantage of this is, if you allow it then to move around avoiding obstacles, and you deliberately break, if something breaks or goes wrong, it relearns. It knows that something's wrong. It can repair itself. And it relearns again over a period of time. But what you see is the frequency come back up again, and it starts to avoid again. You break it again, it'll relearn again. It knows that something's wrong, and then it goes on to fix itself, to re-establish the mapping, the initially learned mapping between the inputs and outputs. That was published back in that paper, 2019. I'm just going to finish off with this. This is a simple little robot, just goes in a straight line, doesn't do big fancy. But I know Jim Harkin, a colleague of mine, was here last year and talked an awful lot about hardware. I won't do that because I probably don't know enough about it, but I can talk about a little bit about this. This is a simple network, 2 neurons on the elasticide. These neurons here have got lots of synapses, and we're going to break some of them. So we took that network, put it down in hardware, put it into that robot, and we let it go off on a straight line, and then we'd break some of the robot with a way of doing that, and it would stop, and then it would repair itself, and then move off again. And you can see here that this is the software simulation compared to the hardware. So as more and more damage is done, 0%, 10, 40, and then 80%, you could recover some of the movement again, but it kind of limped home, if you like. But nevertheless, there was a graceful degradation in this performance, even though you wanted a severe number of faults. I'm conscious of time, so I'm going to finish. So Neuro AI could give us our advances. I'm not saying to stop what we're doing, I'm saying we need to look at this. People need to be trained to this, the link between neuroscience and AI. So it could help us to get closer to human level intelligence, and it certainly will help us to do that with the accepted energy requirements. And that's through, if we use cell diversity, the inclusion of different cell types, including astrocytes and all the different types of neuron cells. There's wiring diversity. I talked before about this network of astrocytes. These sit above the neurons, and they will change the topology, the wiring, over a period of time. If neurons aren't used and pathways aren't used, it'll get rid of them. If it needs new pathways, it'll increase them. So the topology is constantly changing over days, months, weeks, or longer. There's glio-neural interaction. I told you about software repair, and that goes a long way to sustainable systems. And we've done a lot of work with that, both on software and hardware. All this work was done with neuroscientists. So we're trying to stay as biologically faithful as we can. Finally, neural platforms need to be developed with neuroscience, and I'll tell you why. They're going to be the way you can deploy deploy AI because hardware works in parallel like the brain, but also for diseases, neuroscientists want to take, if we can build a reliable model of, say, some sort of disease, Alzheimer's disease, the progression that could be for 10 years. Neuroscientists can't probe a human over 10 years and make some conclusions. But if we can build reliable models, put them in hardware, we can speed them up. We can speed up the progression of these diseases, and then we can look at the triggers for different diseases at different times. Thanks very much.